Active site. The pocket or crevice to which a substrate binds in an enzyme-catalyzed reaction. Amino acid. An organic molecule that has both an amino group and a carboxyl group. Anabolism refers to the metabolic reactions through which cells use energy and small building blocks to build large, complex molecules needed to carry out cell functions and for cell structures. ATP. Adenosine triphosphate, a nucleotide that functions as the universal energy strong molecule in living cells. Carbohydrate. Compounds that contain multiple hydroxyl groups, plus an aldehyde or a ketone functional group, and function in living things to provide immediate and stored energy. Catabolism refers to metabolic reactions that cells undergo to extract energy and chemical building blocks from large, complex biological molecules such as proteins, carbohydrates, lipids, and nucleic acids. Cellular respiration. The process in which glucose is broken down in the presence of oxygen gas to produce carbon dioxide, water and large amounts of energy. Denaturation. The process in which a proton's natura, intricate three-dimensional structure is disrupted. Disaccharide forms when two monosaccharides bond together. Enzyme. A highly specific, powerful biological catalyst. Fatty acid. A long-chain carboxylic acid that usually has between 12 and 24 carbon atoms and can be saturated or unsaturated. Fermentation. The process in which glucose is broken down in the absence of oxygen, producing either ethanol, carbon dioxide, and energy, or lactic acid and energy. Lipid. Large, nonpolar biological molecules that vary in structure, store energy in living organisms, and make up most of the structure of cell membranes. Metabolism. The sum of the many chemical reactions that occur in living cells. Monosaccharide. The simplest carbohydrates, which are aldehydes, are ketones that also have multiple hydroxyl groups, also called simple sugars. Nucleic acid. A nitrogen-containing biological polymer that is involved in the storage and transmission of genetic information. Nucleotide. The monomer that makes up a nucleic acid consists of a nitrogen base and an organic phosphate group, and a 5-carbon monosaccharide sugar. Peptide. A chain of 2 more amino acids links by peptide bonds. Peptide bond. The amide bond that joins 2 amino acids. Phospholipid. A triglyceride in which one of the fatty acids is replaced by a polar phosphate group. Photosynthesis. The complex process that converts energy from sunlight to chemical energy in the bonds of carbohydrates. Polysaccharide. A complex carbohydrate, which is a polymer of simple sugars that contains 12 or more monomer units. Protein. An organic polymer made up of amino acids linked together by peptide bonds that can function as an enzyme, transport important chemical substances, or provide structure in organisms. Saponification. The hydrolysis of the ester bonds of a triglyceride using an aqueous solution of a strong base to form carboxylate salts and glycerol is used to make soaps. Steroid. Lipids that have multiple cyclin rings in their structure. Triglyceride. Forms when 3 fatty acids are bonded to a glycerol backbond through ester bonds, can be either solid or liquid at room temperature. Wax. A type of lipid that is formed by combining a fatty acid with a long-chain alcohol, is made by both plants and animals. Substrate. A reactant in an enzyme-catalyzed reaction that binds to specific sites on enzyme molecules.